Hola, bon dia. En nom de la Societat Catalana de Biologia, us dono la benvinguda a la 19a edició de la Jornada de Virologia. És un plaer comptar amb tots vosaltres. Estem inscrits a aquesta jornada més de 300 persones i esperem que tots vosaltres i les vostres famílies estigueu tot bé, tot i la situació en la que estem vivint. Aquesta jornada la dividirem en dos blocs ben diferenciats. El primer bloc serà en el marc de la recerca del nou coronavirus i serà el Segon Simposi que organitza la Societat Catalana de Biologia. El primer simpòsium va tenir lloc el passat mes de maig, quan es van presentar els projectes que tot just estaven començant. Avui veurem els seus progressos. I el segon bloc el dedicarem als avenços a la recerca d'altres virus, que no deixen de tenir el seu interès i és molt important que es continuï la seva investigació. Good morning. On behalf of the Catalan Society of Biology, I give you the welcome to the 19th Virologia Meeting. It's a pleasure to have all you here. We are more than 300 attendees, and we hope that all you and your relatives are fine during the difficult situation that we are living. This year, we have divided the Virology Meeting in two big blocks. The first one will be dedicated to the new coronavirus through the Second Symposium on Coronavirus Research. The first symposium was in May, when the project on this virus were just starting, and today we will see their progresses. In the second block, we will know the advances on the research of other viruses, which are also important, and investigation is also needed. Però abans de començar aquesta interessant jornada, deixeu-me que us presentem el nou vídeo de la Societat Catalana de Biologia per a que pugueu conèixer la nostra societat una mica millor. But before starting this interesting meeting, let me show you the new video on the Catalan Society of Biology so you can know a little bit more about our society.
La Societat Catalana de Biologia és una societat científica filial de l'Institut d'Estudis Catalans amb més de 100 anys d'història i més de 1.700 socis, que té per objectiu potenciar la recerca que es fa a les terres de llengua i cultura catalanes en el camp de les ciències biològiques i difondre-la a la societat. Es va fundar l'any 1912 amb el nom de Societat de Biologia de Barcelona i actualment té la seu a l'antiga Casa de la Convalescència, seu també de l'Institut d'Estudis Catalans. Tot i que la Guerra Civil i el franquisme en van estroncar l'activitat, l'any 1962 es va reactivar amb el nom de Societat Catalana de Biologia. L'any del seu centenari, el 2012, la Generalitat de Catalunya va reconèixer la tasca de l'SCB atorgant-li la Creu de Sant Jordi.
S'estructura en seccions territorials i seccions especialitzades en diferents àmbits de la biologia. Les seccions organitzen jornades anuals i conferències adreçades tant a la comunitat científica com a la societat en general. Una de les activitats més destacades és la Nit de la Biologia, la celebració anual durant la qual es lliuren els premis SCB. La SCB publica la revista anual Treballs de la Societat Catalana de Biologia des del 1913 i és impulsora d'iniciatives com el projecte Biogenoma i la Bioagenda. La biologia és intrínseca a la societat. Per això, la tasca de la Societat Catalana de Biologia és clau perquè els ciutadans i les ciutadanes tinguin coneixements i criteri en aquesta matèria.
Gràcies per la vostra atenció. Thank you very much for your attention. Avui, doncs, compartirem els nous progressos en el camp de la virologia que ens permetran avançar la nostra recerca per tal de poder transmetre els resultats d'aquesta recerca a la societat. I volem agrair sobretot als ponents convidats que han acceptat participar en aquesta jornada i sé que esteu tots amb una càrrega de treball molt alta i s'agraeix moltíssim que pugueu dedicar part del vostre temps avui amb nosaltres. També volem agrair a tots aquells participants que han volgut compartir la seva recerca amb nosaltres, als assistents el comitè científic assessor, els moderadors, els patrocinadors, sense tots vosaltres, el dia d'avui no seria possible. Today, then, we will learn and share new progresses in the virology field that allow us to advance in our research in order to transmit the results of this research to the society. We are very grateful to the invited speakers who have accepted to participate in this meeting. We know that all you are very busy and we really appreciate that you can dedicate part of your time with us today. We are also very thankful to all the speakers that want to share their research with us, to all the attendees, the scientific committee, the chairmen, and the sponsors. Without all of you today would not be possible. Comencem, doncs, amb la sessió del segon simpòsium de la recerca en coronavirus que la guiaran els moderadors Juana Díez de la Universitat Pompeu Fabra i en Josep Quer de l'Hospital de la Vall d'Hebron. Let's start then with the first session of the second symposium on coronavirus research. This session will be moderated by Juana Díez from the Pompeu Fabra University and Josep Quer from the Vall d'Hebron Hospital. Us dono la paraula. I give you the floor.
Now, so I would like to, it's for me a privilege and an honor to introduce Luis Enjuanes. He's one of my science heroes because of his passion and dedication to science and his achievements. He's a professor at the CSIC and he directs the coronavirus group at the Centro Nacional de Biotecnología in Madrid. Luis is a worldwide recognized expert in coronavirus, doesn't need much introduction, I think, to all of you, but just to emphasize some of the achievements, his team was the first one to develop a reverse genetic system to study coronavirus biology, and also has made great contribution to the virus and host factors involved in the inflammation and all the pulmonary edema related to pathogenic coronaviruses. So besides the undeniable achievements in science, I would like also to emphasize his roles in communicating to science to public in a very humble and realistic way. And also that he is always open for discussion. It's great to talk with him. And he even answers emails very fast in the middle of the COVID crisis. So Luis, thank you very much for taking the time to give this talk. And we are looking forward to your talk.
Thank you, Juana, for the invitation. So should I start sharing the screen? So I will try to summarize. We don't have too much time, so I go straight to the presentation. Coronavirus host interaction and protection. And I will try to summarize what we are doing on vaccine development. So as probably you know, all coronaviruses are classified in 5 different genus, from alpha to gamma. Genus alpha includes coronaviruses infecting domestic animals and men. Genus beta, mice, domestic animals and men again. Genus delta, wild animals and men. Epsilon, bats, and genus gamma, chickens and birds in general. But the key point of this slide is that all these viruses are coming from bats. And these bats are nowadays flying over all continents. Therefore, at any time, a new coronavirus can jump from bats to probably mammals and then to men. Therefore, we need to be prepared for this more than possible event. Coronaviruses, they have the structure that you can see here with the spike protein, which is the major inducer of neutralizing antibodies. And today we know 4 human coronaviruses. The first 4, they are already attenuated and they just cause winter common cold. But the last 3 in red, they are deadly for humans and they include SARS coronavirus that came up in year 2002, MERS coronavirus in 2012, and SARS coronavirus 2 in 2019. And as you can see, they are very different. SARS-1 infected 8,000 people, and whereas MERS, over 17 years, only has infected 2,600 persons, because it only transmits from camels to men, but not very well from men to men. In contrast, SARS coronavirus 2 has already infected more than 44 million people. So the difference are big. And in terms of death, SARS coronavirus killed around 10%, whereas MERS coronavirus was killing almost 36%. In contrast, the present virus is killing around 2%. And they have been infecting 29, 27, and 235 countries. Therefore, it's a huge difference, and the one that is more successful for the moment is SARS coronavirus 2. Now, in this table, I would like to summarize a few events that I consider negative properties of these viruses. The first one is that they cause an asymptomatic infection during almost 14 days. Therefore, they can disseminate, and we don't notice that they are disseminating. They can re-emerge in 40% of apparently recovered patients. So this is probably because the induction of a limited immune response of the virus length. And one of the most important events in these viruses is that they are activated by furin cleavage site on the spike protein that has been recently incorporated to these viruses. I will provide some details on this. If we consider this line, the spike protein is divided in two parts, the S1 towards the amino terminus and the S2 towards the carboxy terminus. And this new virus has incorporated 4 amino acids just in this cleavage site between S1 and S2. But in addition, for infection, the virus needs a second cleavage in S2 primer. These two sites are located on the spike protein in S2 domain, but very close to S1. The S1-S2 site is cleaved by furin even before leaving the infected cells, because the furin is all over basically all tissues. Therefore, when the virus is coming out from the cell, it's already partially activated. But then still, for full infection, it is needed a second cleavage on the S2 primer, and this cleavage in vivo usually is made by tempers or in vivo in tissue culture by cathepsin, another protease. Now, if we consider in the top line the length of the genome, in this position we have the spike protein S1 and S2, and this protein has incorporated these basic amino acids that provide the new activation sites. The terrible thing is that whereas SARS coronavirus 1 only was able to infect the respiratory and enteric tract, this virus, because this site is already pre-activated before leaving the infected cells and can go to any tissue of the body. When we analyze the sequence of different SARS-2 coronaviruses in vivo, it can be found that at least 4.5% of them, they have a deletion on this site with the 4 basic amino acids. Also, there can be deletions more towards the amino terminus of the virus that also inactivate the cleavage site. But this population of the virus that has lost the 4 basic amino acids is not imposed in vivo. It is imposed in certain cell lines like virus cells, changing the behavior in vivo versus in vitro. The consequences are terrible because the virus has a polytropism. It can infect lungs, causing acute respiratory disease syndrome and ending in lung edema. But it can also infect gut, kidney, brain, heart, veins. It can cause the COVID-2, which is due to the bad circulation of the blood, that it can end with strokes and ischemic bowel. Also, it's causing smell loss, and also the keratinocytes are infected because they both, the epithelial cells in the mouth and in the tongue and in the nose, they are loaded with the receptor of the virus, the ACE2. And in severe cases, it can cause also infect pancreas and cause death. So this virus goes much further than the ones we used to know. Among the main characteristics of this virus, as I said before, is also that the immune response has been drastically modified by the virus infection, reducing innate antiviral defenses by causing a reduction in interferon-1 and interferon-3 production. And furthermore, in 10% of the infected patients, the virus induces an autoantibodies specific for interferon-1, which is a disaster. The second major aspect of the virus infection is a huge inflammation due to a cytokine storm, and there are many things that are activated. There are very high levels of cytokine IL-6 that, as you know, promotes the inflammation, high levels of cytokines CXCL9 and CXCL16 that are chemoattractants for T and NK cells, and also high levels of CCL8 and CCL2 that attract monocytes and macrophages. And also the most important cell type for inflammation, there is a high production of CXCL8 that is a chemoattractant of neutrophils, that when they are moving in the blood, they are very peaceful cells, but when they enter in infected lungs, for instance, they destroy not only the virus-infected cells, but also non-infected cells. And another branch of the immune defense are the antibody production. There are many abnormalities in adaptive immune system driven by the dysregulation of cytokine response. Although in many cases, there are similar quantity antibodies are produced in people that survive and those that die. And this is, in terms of quantity, is the same amount, but in terms of quality, in those people that they are convalescent and they survive, most of the antibodies are directed against the spike protein, neutralizing the virus. Whereas in the people that preferentially die, the antibodies recognize mostly the nucleocapsid of the virus. Second problem is the absence of germinal centers. The defect in antibody maturation prevents long-lasting plasma cells production and the production of memory B cells. They are abnormal B cells because they are short life and produce antibodies for a relatively short period of time outside of the germinal centers, and the low neutralizing titer that is observed in those antibodies. So what do we do to fight these events? I will summarize the strategy we follow. Basically, there are 4 approaches that we can stimulate to prevent viral infection. One is natural immunity. The main one is development of vaccines, production of antivirals and antibodies, both plasma from patients that recover and they are loaded with neutralizing antibodies, and also monoclonal antibodies. Now, talking about vaccines, that may take 1 year, but I will simplify that very much. There are essentially 2 types of vaccines, inactivated and live attenuated. The problem with inactivated vaccines is that they induce very low lactogenic and mucosal immunity. In addition, this immune response is non-lasting, has a very reduced memory, and in some cases, they may induce eosinophilia and antibody-dependent enhancement of the infectivity. So they can be useful, but it's not the best choice. The best choice in principle are the live attenuated viruses because they mimic the natural route of infection. They are highly immunogenic. They induce long-lasting memory. Nevertheless, they are not perfect. They have biosafety concerns because we know after many years working with viruses, and you all know too, that we cannot trust viruses because they evolve very fast. And they, even if we attenuate them, they can go back to a virulent phenotype. So when we started the development of vaccines for coronaviruses many years ago, we thought that we should take the new avenues by studying the molecular basis of virus virulence, because contrary to what most people is thinking, viruses, they don't kill because they grow too much. They kill if they have virulence genes. These genes in general, they counteract the immune response, and that is what makes them virulent. Therefore, if we could identify which genes in the genome of the virus they are responsible for virus virulence, we could delete them, the virus will become attenuated, and attenuated virus is a vaccine in principle. Now, to do that, we need to develop two tools, an infectious DNA clone and animal models. And that we did in year 2000. We developed for the first time an infectious DNA clone for SARS coronavirus, and also in collaboration with our American colleagues in the frame of NIH projects, we developed the mouse animal models. Now, having cloned in a bag the full length of the virus, we concentrated our work in the deletion of non-essential genes. They are called non-essential because the virus can still replicate even if we delete them. But we were very brave at that time when we decided the deletion of an structural gene, the envelope gene, which is in a low copy number in the virions, whereas in the infected cells, this is an infected cells, in blue is the nucleus. And when we stain these cells with antibodies specific for the envelope protein, we can see that it's highly abundant. And we were deleting one by one all these proteins, but I will just focus in the deletion on the envelope protein that kept us busy for the last 15 years. The seminal observation was that when we were working with SARS-1 and we deleted the protein and analyzed the virus, the virulence of this virus at 4 and 6 days post-infection, if we were infected the mouse model with the wild type at 4 days post-infection, we could see a strong infiltration, and by day 6, the alveoli is full with water and proteinaceous material, and the animal couldn't breathe and they died. Interestingly, and that was the seminal observation, when we deleted the gene, both at 4 and 6 days post-infections, you can see the lungs are clear, they can interchange oxygen, and the mice survive. Therefore, a Delta virus was an attenuated virus. And then we decided to use this as a vaccine candidate. And after immunizing mice and waiting 3 weeks and performing a challenge of those infected mice, if they were not immunized, they were losing weight rapidly at different time points after challenge, where if we had vaccinated them, they keep the weight constant. Furthermore, when we analyze the lethality by looking survival, we could see that 100% of the mice, they survived. In contrast, the non-immunized one were dead at day 4 post-infection. This observation was made in SARS coronavirus 1, but then we reproduced the same event when MERS coronavirus came in year 2012. In that case, we also deleted all these non-essential genes one by one. I will focus again on Delta E, and we made 2 very important observations. In this case, and in contrast to what happened with SARS coronavirus 1, when we deleted E, the virus could not propagate, was immediately lost in tissue culture during passage. Interestingly, when we look for replication by studying the production of the genome and the subgenomic RNAs, we could see that in black columns, we have here the wild type virus, and in red columns, the Delta E virus. And as you can see, the Delta E virus could replicate more genomes and transcribe more messenger RNAs than even the wild-type virus. Therefore, we have the keys for the development of a vaccine that we think is very safe and strong, because the virus cannot propagate. This is not a virus anymore. This is a replicon. A virus that cannot propagate should be safe because it cannot move from cell to cell. Therefore, it cannot revert to a virulent virus. But it still can amplify the genetic dose that we provide by increasing the genome and the subgenomic RNAs that the virus produces. Now, we analyze the protection within this new system by challenging in mouse that were infected and looking to weight losses and pathogenicity. The non-vaccinated mice rapidly lost weight, and by day 7, all of them die, whereas the vaccinated ones increased the weight and 100% survive. That was an important observation. But to increase the safety of the system, we analyzed the properties of all the other genes. And in summary, I have to summarize that. We observed that by just deleting 4A gene or 4B alone, each one led to an attenuated virus. Therefore, by deleting all these genes, we had at least 3 safety measures for our new RNA replicon based on the deletion of genes 4A, 4B, and E. The virus was replication competent, propagation deficient. But as I said, we should not call him a virus, it's a replicon. In fact, originally we started with the generation of a deletion mutant of E gene, then we removed 2, 4, and 5. And we are working with this as a vaccine candidate, and we have proven that it's very efficient. But we had to make sure that the virus was, in fact, as we were claiming, propagation deficient. Therefore, we study virus titers at days 3 and 6 post-infection. Black columns indicate the growth of the wild-type virus. As you can see, it can produce more than 1 million of infectious particles, whereas the deletion mutant that we engineer cannot grow at all. This is exactly what we want, because it can amplify the antigenic dose, but it doesn't grow. Interestingly, when we analyze by electron microscopy the production of virus, in the wild-type infected cells, you can see in the cytoplasm there are vesicles that are full with virus. But interestingly, in the Delta E virus, we also can see something that is virus-like particles. These virus-like particles are formed, they are highly antigenic because they are polymeric, but they cannot move from one cell to other. But in the vaccine company, we can grow them in large quantities because we can provide E protein and trans and grow that. Note, please, that I am talking about MERS coronavirus. I am referring during all my talk not to SARS coronavirus. For SARS coronavirus 2, we have done exactly the same thing, but the conditions are different. The genes that you have to delete are different. You have to identify specific combinations of genes to engineer a replication competent, propagation deficient virus. We check the attenuation of this RNA replicon, and all the forms, all the replicons that we engineer, they cause no weight losses in contrast to the non-infected mice, to the mice already infected with the wild-type virus. And when we check for pathogenicity, also all mice infected with all these RNA replicons, they could survive 100%, in contrast to the control. All the mice were dead by day 8. And when we check for protection, when we immunize mice and 3 weeks later, we perform the challenge, the non-immunized mice, they lose weight and die, all of them by day 7. In contrast, the vaccinated animals, they increase weight and survival is 100%. Interestingly, our system is causing what is called sterilizing immunity. What does it mean? Does it mean that if we immunize mice and we perform a challenge and we look for the production of the challenge virus, I have to tell you that we provide to the mice more than 10 to the 5th infectious doses. Look what happens at days 2, 4, and 6, the virus is not growing at all, in contrast to the growth in the non-vaccinated mice, black columns, that they can produce more than 1 million infectious particles. This is called sterilizing immunity. And now we are making, I have to summarize that a lot, 2 versions of the vaccine. One is a chemically synthesized version. This version is based in the use of the RNA replicon that we transcribe in vitro efficiently. We transcribe 28,000 nucleotides very efficiently with high yield, but we have to wrap up this RNA replicon with a cationic polymer for 2 things: to protect the RNA from degradation and to form nanoparticles that will penetrate the cells efficiently. Therefore, this is fully chemically synthesized, and I think that it will be very efficient. We know that it's efficient when we made experiments with MERS coronavirus 2. There's a second version, is the use of virus-like particles, the ones I told you. We produce them by transfecting cells with the replicon and provided in trans a specific combination, in the case of SARS coronavirus 2, of proteins that are needed for propagation. As I showed before, that can form VLP particles that can provide full protection, sterilizing immunity. And I don't know how I am doing. If I have 3-4 minutes, additional minutes, I summarize at least one part on how we have been developing antivirals. When we have a virus infection, usually, certainly when we infect with a coronavirus, we induce inflammation, lung inflammation. And this lung inflammation can be produced through 5 different signaling pathways. By doing studies analyzing which protein has been promoted in the nucleus of the cell, we identify that by infecting with SARS coronavirus 1, but the same is happening with the other coronaviruses, what we are stimulating is NF-κB. And therefore, we thought, well, if the only thing that is differentially stimulated in our attenuated virus, because we deleted the protein, if we inhibit the signaling pathway, the stimulation of NF-κB, probably we can save lives. And that was the case. We used 2 inhibitors, and the combination of these 2 inhibitors, in the first case, red columns, we use no inhibitor, and we analyzed the production of pro-inflammatory cytokines like TNF-alpha, CCL2, CXCL2, and in inhibiting those cytokines, inhibiting the NF-κB signaling pathway, we inhibited the production of these pro-inflammatory cytokines. And that was not due to the reduction of virus titers, because as you can see in this slide, they are produced to the same titers, but ones they cause the death of the animal, whereas the others they cause no disease. Let me summarize the last piece of information I am trying to present. E protein makes self-aggregates, it makes pentamines, it's a transmembrane protein. And at the carboxy terminus of this protein, there is a motif that we have been studying for more than 10 years now. It's a motif, a virulence motif, very strong virulence motif, because it has the power to bind another motif, PDC, which is present in mammalian cells. But the relevant issue is that PDC motif is present in more than 400 cellular proteins, mammalian proteins. Therefore, just the envelope protein of the virus will be able to bind to more than 400 cell proteins and control the cellular metabolism. This motif, the PDC domain, is a protein recognition sequence that consists in 80-90 amino acids. And there are more than 400 cell proteins that contain this motif that can be seen by the PBM of the virus. To analyze the role very briefly of this motif, we engineer 4 coronaviruses. 2 of them, the first and the last, will have the PBM motif, whereas the other 2, in one we will delete the sequence or just mutate them by glycines. When we analyze the virulence we infect with those mutants, different humanized mice, and checking the lungs, the consequences of this infection, if we do not infect the mice, this is the lung. It's very clean and functional, it can interchange oxygen, but if we delete the PBM from a virulent virus, still the lung is functional, it can interchange oxygen. But if we use the wild type virus, look what happens 3-6 days later. There's a huge infiltration and lung edema. The alveoli are full with water. They cannot breathe and die. Therefore, the conclusion is that this PBM motif is a virulence factor. And as I don't have time, I will summarize the work of 3-4 years in one slide. What we had observed by using system biology is that the SARS-CoV-2 protein was binding synthenin. And synthenin is a cellular protein which is part of the extracellular matrix complex. And what this complex does is to promote the phosphorylation of P38 MAP kinase, and that will stimulate NF-kappa B and an inflammatory response. So that was the proposal. But along many years, we demonstrated that this step was taking place, that one and the other one. We demonstrated the mechanism. And we thought, well, fantastic. We have discovered a signaling pathway that is activated by the infection of coronaviruses, particularly the deadly ones. But there are many signaling pathways in the cells. How important is the signaling pathways we have identified? If that will be very important, if we would inhibit P38 MAP kinase activation, we should prevent lives. And that is exactly what happened in just the very first experiment. If we were infecting mice with the virus, the wild type virus, by day 5, survival will be 0. All them were dead. In contrast, if we added to the system inhibitors of P38 MAP kinase, 80% survive. Just with one inhibitor of the P38 MAP kinase activation. There are more than 800 activators in the market because they are heavily used in cancer research. So in conclusion, the PBM of SARS-CoV-2 protein is a determinant of pathogenesis, a fantastic determinant. That means presence of PBM is death or life. And we have identified efficient antivirals, which are all those that inhibit the activation of P38 MAP kinase. And I think I have to end here. I don't know, I lost control of the time. Just to mention some of the people that have been helping a lot in SARS-CoV, Jose Manuel Onrubia, in SARS-CoV-2, Javier Gutierrez, with other virus system, Sonia Zúñiga, and I am co-directing the lab with the help of Isabel Sola. We have collaboration for the last 15-20 years with American groups under the umbrella of NIH projects. And also we have EU projects. These efforts have been devoted mostly to the production of monoclonal antibodies against all these viruses, including SARS-2. We already have neutralizing antibodies, and we are running those antibodies in clinical trials, phase 1. We have also been collaborating with Adolfo Garcia Sastre, and I didn't have time to talk about other antivirals that using our system in good collaborators from CNRS in France, Jean-Paul Borg and Pascal Zimmermann, they are producing a few molecules that we are testing in our laboratory. Thank you very much for your attention, and I am open for your questions if there is any time left.
Thank you for this great talk. I forgot to remind you that there's in the down of your screen, you have a question and answer, and you can put there your. So I have a few questions for you because, well, we have a lot of questions, but not so much time. So, Luis, you mentioned that the disease-infected patient had more antibodies against nucleocapsid. This is a very intriguing issue. Is that a sign of a high T-cell response that is detrimental for the patients?
I am not sure I got your question, Juana. You are talking about the immune response, the antibody response?
Yeah, because there is a difference, no, of the quality of the antibody response that.
Oh, yes.
And the other no, and if the antibody response against the nucleocapsid that happens in people.
Correct.
Is a sign of high T-cell responses that could be detrimental some way for the patient.
Well, we believe for the moment that in principle, the T-cell responses in general, they are helpful. Furthermore, it has been speculated, but at least by now there are 3-4 papers, including one by my friend Christian Drosten from Berlin, that saying that from the 7 human coronaviruses that we know today, the 4 old ones that today are already attenuated, they induce when they infect all human beings, basically. And when they induce human beings, they induce T cells that still can cross-react with the present human coronavirus, with SARS-2, with SARS-1, and with the MERS coronavirus, and that they provide some protection. In fact, they have proposed that there are so many asymptomatic patients because many people has been infected by these coronaviruses that just cause the common cold, and they can protect us. So in summary, there are we could consider 2 types of T-cell responses. The one induced by the past coronaviruses that do not cross-react serologically with antibodies with the present one, but that they induce T-cells that has been demonstrated by European and North American laboratories that they cross-react with SARS-CoV-2 and can provide protection. But in addition, as you know, SARS-CoV-2 also induce good T-cell responses, and in fact, we are characterizing those T-cell responses in our vaccines. So I think T-cell responses are essential. The main thing to protect against coronaviruses are the antibodies. They do majority of the job by neutralizing all the antibodies. But the left viruses, those that can be escaping mutants, these are the T cells who have to take care of them.
We have here another question from Albert Bosch. How many doses of your vaccine you think it will require for successful response, and how far are they to be used in humans?
Well, I should say, and I think this is the truth, our vaccine, as I mentioned before, is sterilizing. That means that the immune response that is inducing is very strong. From the vaccines that are now highly advanced, only 1 out of 7 is able to induce sterilizing immunity. And why is that? For a main reason is within the textbooks, all we know that to induce a strong mucosal immunity, you need to induce locally because IM immunization, as everybody knows, is not able to induce a strong and long-lasting immune response. In contrast, in our case, we have chosen intranasal immunization. Therefore, the virus by instillation, the virus goes straight to the lungs, to the right place to induce a strong mucosal immunity. In addition, remember that our virus is based on an RNA replicon, which contains many viral proteins. We just only remove 5 of them, but all the others are expressed by the RNA replicon and in the right place, the lungs. Therefore, we are not surprised if in all cases we already performed many experiments, particularly with MERS coronavirus, and in all cases we have sterilizing immunity. And I can guarantee you that this is very unusual today, even with the best vaccines, including the Oxford vaccine or Moderna vaccine and many others. There is only one additional exception.
And do you escape the part of by when do you think this will go clinical trials and have some results?
Whereas I claim always that our Whereas I claim always that our vaccine should be highly protective, we are more slow than the rest of the people. You have to take into account that most of the laboratories in the world, they are using already very well-known vectors based in adenoviruses that are known for many, many years, or vectors based in pox viruses that already approved in all the world. Our vector is new, and therefore we have to prove that it will not induce secondary effects. This is our main limitation. And whereas everyone is claiming that the vaccine will be ready by the end of this year, or I don't believe it, but it will be probably ready in the first or second trimester of next year. Ours will not be ready until the end of next year if everything goes well.
Okay, and then a final question that they say, if you use inhibitors of the inflammation, how is the response of the virus affected, especially the protective response?
Well, the response to the virus in the situation we are approaching, this is a very good question, I think. In the situation we are approaching with a cytokine storm, even if we inhibit the pro-inflammatory route, still the immune response will be working out, because what we have in the patients that have been deeply infected is a cytokine storm. Therefore, even if we immune suppress inflammation, still they will have the power to mount a strong immune response, both humoral and cellular. I will not worry about that.
Okay, thank you very much. We'll have other questions, but there's no more time. Thank you again for connecting with us, and we are really looking forward to your results. In the
Thank you, Juana. Thank you, all of you.
Bye.
Okay, bye.
Okay, so thank you very much for the outstanding talk, Luis. And let's move to the next talk. For me, it's a great pleasure to introduce our next invited speaker, Dr. Júlia Vergara. She's a veterinarian and researcher in the coronavirus group at CReSA IRTA. She did several stages in the United States, Italy, and a postdoc in Germany. And for the past 10 years, she has focused her research on emerging zoonotic disease, from influenza virus to the highly pathogenic coronavirus, such as MERS. And she has a vast experience in animal models, using from camels and fire pans to mice, and working in maximum biocontainment laboratories. But lately, she's an expert journalist collaborating in the Telenoticias Vespre de BTV each Monday, and also she's a Twitter influencer with almost 1,000 followers. And recently, she has been incorporated into a staff of the experts in the World Health Organization, this group is looking for animal models to reproduce the COVID-19 disease as a model for antivirals and vaccine development. So the best thing that we can do is just to listen to Júlia, which is going to talk about advances in animal models for the study of SARS-CoV-2. So please, Júlia, share your screen, and up to you.
Okay. Well, thank you very much for this complete introduction, Josep. It's really a pleasure to be here. Of course, I would prefer another kind of situation, but we need to adapt to these times, and we are discussing about SARS-CoV-2. So I guess these teleconferences are more and more usual. So as already Josep has introduced, I will talk about advances in animal models for the use of SARS-CoV-2 studies. And also, as Josep said, together with Kim Segales, I am a member of this COVID-19 animal model working group in the WHO since last April. And well, here on the right side of the screen, I'm not sure if you can see my mouse. So this is the latest article we published all together with all the, well, not all the members, but some of the members of this working group, summarizing the animal models for COVID-19. And the great thing of being part of this working group is that, well, the main objective now is that we want to standardize mainly the animal models that we are going to use, mainly to evaluate vaccines and treatments and their efficacy, of course, and also to study, to study the enhancement. This is a really important topic that we discuss weekly. The enhanced disease after vaccination. Of course, the animal models are also being used to study the pathogenesis of the disease. So the main task that we are doing is discuss the recent results. So as you all know, results are well, all the laboratories are obtaining results very fast, but sometimes to share these results is really difficult. So weekly we share our recent results, and then of course we share the protocols, and always we are in constant review. So what we pretend is to share the information, but also the capacity worldwide to conduct all these animal studies. So starting with the first animal model that I wanted to talk about is the laboratory mice. As you probably all know, this new coronavirus, SARS-CoV-2, does not infect the wild-type laboratory mice, let's say. But of course, some strategies have been developed to obtain susceptible mice. Some of them, as Luis also mentioned, have been already developed for other coronaviruses. So what we can do is to modify the mice, to express the human ACE2, that is the receptor, via different strategies, transgenic mice, of course, knock-in mice, and also some virus that virus-based delivery, that we can give this human ACE2. Also, the virus can be adapted to infect the wild-type mice, let's say, as also Luis has explained, and they have a lot of experience on that. What is the problem about this? That the infection is really severe, but is not does not fully recapitulate what happened in humans. Here is a short summary of what we obtain after infecting mice, and the dose that we have been used is more or less 10 to the 3 or 10 to the 40 CID50s per animal via intranasal instillation. And we can observe, well, yeah, this is from others researchers. But what we also observe is, of course, loss of weight of these animals, but importantly, and besides respiratory clinical signs or lesions such as pneumonia, the virus also affects in the brain. So, and in some cases, animals die because of these neurological signs. So the lesions are really severe. So in summary, this model is, as I was talking about, so we have several mouse models that cause between mild and severe COVID-19, let's say, lesions. That we don't have nowadays a mouse model that recapitulates all the aspects of COVID-19 in humans, but that's true that we can use some of these models, different models, and depends on our objectives to evaluate vaccines and antivirals, because that's true that some models share some aspects as in human disease. Another animal model that I wanted to talk about is hamsters. Golden Syrian hamsters have been used a lot, I would say, and I would say that this is a better model than mice because hamsters suffer mild to moderate disease. We can detect weight loss, as you can see here. These are two different graphs, but showing the same. The animals lose weight, and the only difference is that in the left side, the left graph, you can see the typical curves of young hamsters, and on the right side of adult hamsters. But in both, we can detect loss of weight. In this model, we also see virus replication in both upper and lower respiratory tract, and also pulmonary pathology. Moreover, some authors reported that the infections in hamsters reflect to some extent to demographic differences that have been observed in humans. For example, the male and older animals or humans, let's say, in both, the disease is more severe. Some transmission studies have also been performed in hamsters, and has been observed that this transmission can occur. Here, this graph is just to show you that these are animals that were not receiving the virus, but were in contact with infected ones. And here we detect both on the right side weight loss, and on the left side virus and the respiratory tract. So this model has been used to test both vaccines, but also therapeutics as hydroxychloroquine, favipiravir, and in fact, in this model, these antivirals have shown limited or no efficacy also. Well, I invite you to hear the flash talk of our postdoc fellowship, Marco Brustolin. He will present also a recent study that we want to publish soon, that it's about if hamsters, can be reinfected or not with SARS-CoV-2. So in, I don't know, in 1 hour, something like this, he will present this work. What about ferrets? Ferrets have been used to study several respiratory diseases, and of course, also been tried to study the SARS-CoV-2. In ferrets, no clinical alteration or mild alterations have been shown after infection, but it's true that viral replication occurs in respiratory tracts and also in gastrointestinal tract, and that the virus can be shed from the upper respiratory tract, we can say in a long-term way, up to 2 weeks. Moreover, also transmission studies have been done in this animal model and proved, and also the airborne transmission has been observed. And these graphs here is just to show you that the viral RNA, but also infectious virus, have been detected in the contact animals, the non-infected one, but the ones that were put in contact with the infected ones. And of course, non-human primates have also been used. We all know the problems with working with this animal model, ethical problems, and of course it's difficult, but they also give some benefits. And for example, high levels of virus have been found in both the upper and the lower respiratory tracts, and pneumonia has been also reported. Also, some studies of rechallenge have been done, and some of these animal models have been shown to have a high immune responses after the first infection. That means that they were resistant to a rechallenge. As in hamsters, some demographic differences have been shown between elderly animals, that they show severe, let's say severe disease. And of course, as you all know, vaccine studies, but also therapeutic studies are being doing in these animal models. here, that's true that we really need to be really carefully because some of these models, depending on which non-human primate you are using, you can have slight differences in terms of severity, for example, or shedding. And then there are other animal models that have been done. For example, mink, cats, dogs, pigs, chicken, ducks, fruit bats. And also important here is, and I will explain you in the next slide about the reverse zoonosis. And here, for example, I highlighted pigs because at IRTA-CReSA, we worked with pigs. We had evidence that pigs might be susceptible to infection because of some in silico studies that we did and others that were appearing. But well, as you can see, once we inoculated pigs, and we did it by different routes, by intranasally, intratracheally, intramuscularly, and also intravenously routes, we observed that they were not susceptible to infection. But that's true that the ones that were inoculated, so the groups that were inoculated via intramuscularly and also intravenously, they had some kind of immunogenicity, and here is what I show in this graph. So on the right side are the animals that were intramuscularly vaccinated and not vaccinated, sorry, challenged, and also intravenously challenged. And also, and just to mention that, well, there are several studies showing experimentally that cats can be infected with SARS-CoV-2, and they can also transmit the virus to those cats that are non-infected. But here, and maybe you already heard that at CReSA also, we did a study with a cat and we detect the SARS-CoV-2 in a cat that was owned by a patient, a COVID-19 patient. And this cat showed viral RNA in nasal turbinates, in nasal swabs, and also in mesenteric lymph node. But that's true that we conclude that this was a reverse zoonosis. But and the animal died, but it died for another thing, for cardiomyopathy. And here on the right side, what I show you is the immunological outcomes. So this cat developed antibodies against SARS-CoV-2. So in fact, this cat was, we proved that it was infected by coronaviruses, but we still kept that this was a reverse zoonosis. And this is a summary of the different different animal models, and compare it to humans, Because sometimes, and depending on what we want to study, we may choose one animal model or another. And for example, if we see here on the table on the left, in terms of virus replication, for example, we can find the virus in the upper and in the lower respiratory tract in some animal models, as it happens in humans, such as mice, ferrets, mink, cats, and bats. And then in the lower, as I told you, hamster recapitulates really good what happens in the lower respiratory tract in humans. But then, for example, well, in fact, here, as I was mentioning, pneumonia. So hamsters, together with non-human primates, are one of the best animal models to recapitulate this pneumonia in humans. And then if, for example, we want to see some immunological outcomes, there are some animal models that seroconvert, and this has been proved by several groups, such as different models of mice, hamsters, ferrets, and of course, non-human primates and bats. And just to sum up a little bit, what are we doing at CReSA? Well, I told you about the reinfection studies that Marco will briefly explain later. We have been working hard to have a model for SARS coronavirus 2, and also I think that it's a good model to study transmission. If we want to study vaccines, it's important to, for example, if we are thinking about vaccines, it's important to decrease or to stop all the clinical signs, but also if, as Luis was mentioning, a sterilizing vaccine, if we can stop the transmission, this is the best vaccine. So having a model that can also mimic the transmission, this is great. And then also not antivirals and vaccines, but also to test antibodies, of course. And currently, what we are mainly working at CReSA with golden Syrian hamsters and transgenic mice, but we also have other models of mice in development. And then, well, this large animal model in swines that in fact we are also doing a study of immunogenicity, because as I told you, as they have some antibody responses after intravenously or intramuscularly administration, we can do some immunogenicity studies. And then, well, as you know, there are other models that have been tried and to avoid animal experimentation, some microfluidic tissues from humans have been used, ex vivo explants, and all others that have been deeply used. And of course, they cannot substitute the final testing in animal models, but we can have a lot of information using them. And some recent updates about the animal models and results that have been generated. For example, that using convalescent serum can reduce the severity of COVID-19, and this has been approved in non-human primates. Also that in hamster model, in this case, neutralizing antibodies, well, has been used as also as a therapy with good results. And then that also, even if the transgenic mice that I was talking about here is one concrete transgenic mice, that they are showing severe respiratory disease, but they are being using to study other pathogenesis such as anosmia, that we know that is one of the clinical signs observed also in patients. And that's all from my side. I want to thank all the consortia that we are involved with. So IRTA-CReSA, together with Barcelona Supercomputing Center and IRSI-CAIXA, we are working together since several months ago. And we are also funded by Jaume Corono, and also by la Generalitat de Catalunya. And that's it. I'm open to questions.
Thank you very much, Julia. We have time for 2 questions. There are no questions in the audience, so let me ask you, if you have to choose now from your point of view, which is the best model to study, animal model to study vaccines? Which one will you choose?
To us, as we were mentioning, golden Syrian hamsters is one of the best model. But of course, I'm also in the opinion that when you have a candidate, one thing is to try, if it works or not, but it's always good to have at least 2 models and to prove either your antiviral or your vaccine in at least 2 models. But if we need to choose one for everything, let's say, because it's more practical, for example, than a human primate and all the ethical concerns, and not all the labs can work. We cannot work, for example, with non-human primates, and all the similarities that hamsters has with humans in terms of COVID-19, of course. I think that we will stay for it.
Okay, and last question from Fernando. DCAM peak alveolar macrophages can be infected by COVID-19 in vitro? The macrophages of the DCAM peak alveolar macrophages. Do you know that?
I'm not really sure about peak, you said, or not?
Yeah, peak.
Yeah, several studies have been done in this, so because I cannot see the question, so it's from pig.
Yeah,
okay.
From pig.
When we went to study pigs, it was, as I told you, because there were evidence in silico. So we, but also others, together with our colleagues from bioinformatics, predict that pigs, and in fact also some avian species that can be infected. And there was some evidences in some pig cell lines. I am not sure if in macrophage alveolats this have been proven or not. So I don't know.
Okay. And last question, because we had no time, and there is by Jordi Rambla, there is any hypothesis for the differences between the models?
Between the models, so all the models, you mean? Yeah, of course. So one of the main difference is the receptor. So the susceptibility of the receptors, depending on the different animal models, is different. No, the different amino acid of this receptor. This is one of the main difference. Of course, for example, for hamsters, the receptor is very similar as for humans. So and also non-human primates. So this is one. And for example, for mice, the receptor, the ACE2 receptor, is quite different, and in particular in some parts. Some very important parts of this receptor for the binding of the virus, it's different. So this is one of the main difference and main reasons of that.
Okay, well, thank you very much. I received the comment that we are very late in the time. So, so if you have any other question, you can send it directly or put it into questions and answers, and we will answer you. So thank you very much, Julia. And we have to move to the oral presentations. So Juana, your turn.
Yeah, now we will start a section of short talks. So the first, I will remind you that you can ask your question to the question and answer part. Don't forget that. And I will also like to remind the different presenters to try to get on time. They have 10 minutes and 5 minutes for questions. So the first presentation is SARS-CoV-2 induces cytokine release by human macrophages in the absence of productive viral replication, presented by Daniel Pérez-Solt from the IRTA-CReSA in Badalona. Thank you, Daniel.
Hello, everyone. So first of all, I'd like to thank the organizers of this meeting for choosing this presentation, in which I will share some of the results that we obtained in our laboratory at IRTA-CReSA, and so we can discuss them all together. In particular, I'm going to talk about how SARS-CoV-2 induces the cytokine secretion by human macrophages in the absence of productive replication. As an introduction, I'd like to remind the natural course of SARS-CoV-2 infection, which consists on several stages. In the early stages of the disease, there is a high viral replication, but as the disease progresses, there is a host inflammatory response that may cause the acute respiratory distress syndrome, a common complication of the disease that is associated with mortality. In order to treat the infection, the potential therapies should include antivirals to control viral replication at the early stages, but also immune modulators to control the inflammatory response that we observe at the later stages. However, in order to design the best therapies, we need to understand the insights of viral replication to design antivirals, and also the biology behind this inflammation. Myeloid antigen presenting cells, such as macrophages and dendritic cells, are key players in immunity because they initiate both innate and adaptive immune responses. However, to this date, we don't know many important features regarding the interaction of these cells with SARS-CoV-2. For example, we don't know if they can support viral replication, and it is also uncertain whether they can produce inflammatory cytokines in response to the virus. So here we assessed, first of all, whether SARS-CoV-2 can replicate in macrophages and in dendritic cells. For this purpose, we incubated the cells with SARS-CoV-2, and after 24 and 48 hours, we assessed we measured the viral content of the cell lysates and also in the supernatants. As you can see here, when we assess the cell lysates, we observe that macrophages are associated with high amounts of virus at early time points, but then this content diminishes. This is possibly due to the capacity of these cells to capture the virus, but then also to degrade it. In dendritic cells, we don't observe high levels of virus, but we speculate that something similar might happen at earlier time points. I mean that these cells can also capture and degrade the virus. However, regarding the first question of this experiment about the viral replication, in case there was, we would expect an increase in the viral content over time, which we do not observe in this case. When we assessed the supernatants, we didn't observe this viral increase in any case. So the conclusion of this first part is that our data suggests that macrophages and dendritic cells do not sustain productive viral replication. Moving forward, we assessed if these cells can produce pro-inflammatory cytokines in response to the virus. For this, we incubated the cells with the virus, and in this case, aside from macrophages and dendritic cells, we included a positive control that consists on dendritic cells exposed to bacterial lipopolysaccharide, or LPS. This is a positive control because we know that these cells produce cytokines regardless of the virus. So after 24 and 48 hours of culture, we collected the supernatants and assessed the cytokine content by Luminex. As you can see here, we obtained these results in which, for example, for IL-6, we observe that in presence of the virus, macrophages, but not dendritic cells, are able to produce this cytokine. Similar results were obtained for the pro-inflammatory cytokine TNF-alpha and also for MIP-1-alpha. Another pro-inflammatory cytokine, IP-10, was also produced by macrophages that were exposed to the virus, while these levels were lower in the supernatants of dendritic cells. So, as we observed this production of cytokines by macrophages, we then asked whether we could block this cytokine release by macrophages exposed to SARS-CoV-2. For this purpose, we incubated again the cells with the virus, but in this case, we added immunomodulators, and we assessed the cytokine presence in the supernatants by Luminex. What we observed is that in the presence of the immunomodulator, cells that were exposed to the virus had a lower amount of IL-6 in the supernatant. In the case of IL-6, reduction was by 36%, while in the case of TNF-alpha, we observed a reduction of 42%. For MIB1-alpha, the differences were not statistically significant. However, the reduction was about 90%, and for IP10, the reduction reached 67%. So, as a final conclusion for this talk, we observed that among myeloid antigen-presenting cells, macrophages but not dendritic cells are able to interact with the virus, and while this interaction did not lead to a productive viral replication and thus antivirals would not act at this level, we indeed observed a production of pro-inflammatory cytokines, which could eventually lead to the acute respiratory distress syndrome in these patients. However, we observed that with immunomodulators, we can reduce the secretion of these cytokines. We also think that we generated a platform that would be useful for testing the efficacy of other immunomodulators that could be further used in clinical settings. So finally, I'd like to acknowledge all the people involved in this work, in which people from IRTA-CReSA and the Hospital Germans Trias i Pujol have participated, but also with a crucial collaboration from people from IRTA-CReSA and from the Barcelona Supercomputing Center. And now I am open to any questions you may have. Thank you very much.
Thank you, Daniel, for sharing your interesting results with us. I have a question. So what part of the virus do you think might be responsible for the induction of the inflammatory response?
Well, we didn't assess this part at a molecular level, but for what we know about other viruses, the viral RNA might be having a sensing in the cytoplasm of the cells through several TLRs. But that's a question that remains open and we should address.
Here there's another question from Matilde. So human macrophages are bone marrow derived macrophages, M1 or M2?
So we have a model in which we derive the macrophages from monocytes from the patients and are not bone marrow derived. And either macrophages and dendritic cells are monocyte derived. And they are the kind of inflammatory macrophages.
Okay, thank you very much. And we will pass to the next talk. Thank you, Daniel.
Okay, so let's move to the next talk, which will be done by Marc Martí Renom from CNB-CSIC. And the title of the talk is Tissue-Specific 3D Genome Conformation Predicts Novel Genetic Susceptibility Lossy for COVID-19. So, Daniel.
Gràcies, Josep. Em sentiu bé?
Yes.
Okay, good. I'm going to share my screen, and while I share it, I thank also Nuria and Juana for putting this together with Josep and many others. My talk is going to be about what we call tissue-aware GWAS. So what we want to do here is to analyze GWAS data, recently released GWAS data from COVID patients, to see whether we have some interesting targets to detect based on what we call tissue-specific 3D genome conformations. I'm going to try to explain you what that means in a second. Now, I changed the title of my talk, and I included severe there, just because that the field in GWAS in COVID is moving very rapidly, and only a few days ago, we got a very good data set that compares patients that end up with severe COVID compared to patients with COVID, but that do not have severe COVID. So the idea is very simple. What we want to do is right here. What we want to figure it out is whether when you do this GWAS analysis, you compare the cases and controls. And in this case, all these are cases which each one of them have a particular mutation or variant in different color. And none of the variants is very much significantly over represented in the cases control compared to the controls. But what if these mutations end up especially close in the nucleus? What that means, we do have 2 meters of DNA in each nucleus in our cells, and these 2 meters of DNA need to fold and pack within the nucleus. That means that there are certain regions of the genome that get closer in the space. And the question is, do we have this environment in three-dimensional space that is enriched in mutations in these cases? Why do we think that this could be the case? And in fact, our lab has been working in three-dimensional genomics for many, many years. The first observation is this what you see here that we published last year with Jorge Ferrer in Nature, where we show clearly the existence of what we call three-dimensional hubs of enhancers. What you have here is a part of the genome represented in 3D. In blue, you have the promoters, and in red, you have enhancers around it. This could be somehow a three-dimensional super enhancer. Now, is this true? And we believe it's true. What happens if you have several mutations in these hubs? Would they affect the expression of the genes, even though the genes might not be mutated themselves? And the first observation that we made relating GWAS to these three-dimensional aspects is in this plot here. What you have here is in the y-axis, you have the p-value, the negative log of the p-value of the GWAS. Each one of these points is one SNP or one variant. And what you can see here is these 3 peaks. None of them is really significant. So they would not account as a tower in the Manhattan plot, but they have come together in a space. And what you have up here is what we call a high C. And this high C is basically the interaction between these 3 points. So if it's very red, it means that 2 points in the genome, and these particular 2 regions of the genome in chromosome 20, interact very often to each other. And you can see that 3 of them come together in the space. So this would be a hub of relatively enriched mutations in a particular disease. So do they exist? And if they exist, are they affecting COVID, and how we can assess this? So we developed something that we call meta-loci. It's a computational method developed by Juan. In fact, Juan had to deliver today the talk, but unfortunately he had to be traveling, so I'm delivering on his behalf. Juan is a postdoctoral fellow in our lab that together with Oscar Lau at SENAC, we all came up together with this type of method, and we are using data sets that are derived from cells that are relevant to COVID by Ralph Stadhoders, Greg Stick, and Thomas Graf, the last 2 at the CRG, and the first one, Ralph, at Erasmus MC in Holland. So what type of information do we need to make these assessments? We need 2 types of information. One of them is the 3-dimensional interaction maps of these cell types that are of interest, and I'll tell you later on which cell types we choose. And the other type of information that we need is GWAS analysis, what is called the summary statistics that tell us which particular mutations are more relevant to the disease of interest. We put those 2 types of information in our computational method, meta-loci, and we build these 3-dimensional or 2-dimensional hubs that we call meta-loci. And I'm going to try to walk you very quickly through that because of the time constraints that we have. But if you have questions of the method itself, just ask me later on, even in private if you want to. So the first set of data is this COVID GWAS analysis. We have several of them since the middle of the summer. The first one was published in this New England Journal of Medicine, almost in parallel or concomitant with this other one here from the COVID-19 Host Genetics Initiative. You can get the data here. It's a consortium of many, many groups and labs across the world. Both of them identify very clearly this peak in chromosome 3, this tower in the Manhattan plot. Again, the log of the p-value of the thing. and each one of the SNPs, and they identify this cluster of genes around this tower that are related to a certain extent to cytokine storm. In that paper in June, they also identified the ABO locus here, but that has not been confirmed later on. So this is what we had in summer. We started working on this data set, but then only recently, and that's about 17 days ago, they released the October 2020 data set. The first thing that we can see here is that in this particular subset of the GWAS analysis, where they have about 8,000 cases against 1.7 million controls, that's the population in general, they really confirm the peak, very extremely significant that it was found before. Now we're talking about 10 to the minus 70 in p-value, very, very significant. They find additional ones around here. By the way, this line here sets 10 to the minus 8, which is normally the what we have here, the number. So the significant threshold. But then they also release a data set that in our case is much more interesting, which we have severe COVID patients, about 2,400 of them, versus people who got COVID, but they didn't end up with a severe version of the disease. You can see that the Manhattan peak here is almost significant, 10 to the minus 8, remember? But it's there. You can start seeing it even with only this little amount of cases and controls compared to this one here. So we decided to work with that one here, the October 2020. Which cells we choose? We choose 7 cell types to analyze three-dimensional conformation: epithelial and endothelial, and then B cell, blood cells such as B cells, macrophages, neutrophils, and 2 types of T cells, CD4 positive and CD8 positive, because they are all somehow related to the susceptibility and to the infection by COVID, and we have heard many things of this. We get the high-C data, and these are some numbers that are not relevant for the sake of time, but these are the amount of interactions that we get in the genome. The first thing is that we divide the genome into linkages equilibrium blocks. So we take the entire genome, we divide it into pieces, and these are in chromosome 20, we get about 1,700 of them, and these are very small pieces of DNA, each one of them in principle in linkages equilibrium between them, so there is no correlations of the mutations. Then we ask ourselves, are these blocks in the genome, and that will represent one part of the genome, and you can see the variability on sizes of these blocks. Do they interact in 3D? That's a question. And we use pieces of work that we developed recently to identify which parts of the genome, and this will be each one of these squares will be one block, which parts interact more often to the others beyond randomness. And every time you see a red part here, it means that they do interact more than expectations given the genomic distance between the blocks. So the next thing that we do is we take these interactions that are significant and we build a 2D version of this, where each one of these dots is one LD block, and then if they get close, it's because they interact. So for example, the interactions that you see here in this sort of half triangle or triangle, which by the way, in our field is called topologically associating domains, you can see here that they come close in the space. So we find this Bode-Feder representation in 2D of the interactions that happen in 3D in the genome, and this is a particular region of chromosome 1. Then we go the GWAS analysis, and we get each one of the variants, and we have what is called the odds ratio of the variants. The higher this odds ratio, the more differences between control and cases. And we throw this value, which is called the tree mean, into this map. And we find that in the map, sometimes you get cluster of high values of odds ratio in all the SNPs within each one of the blocks. And there are areas that have low values. You can see that this is not totally random. If it would be random, all the dots would be more or less the same values or scattered distributed, but it's not. In fact, we have an algorithm that detects it's what we call hotspots of variants. So we have a lot of variability happening here between cases and control. And then we use local Moran index to identify the metallocytes. And this is really the metallocyte. What is a metallocyte? It's composed by linkage disequilibrium blocks that we call seeds and another ones that we call neighborhoods. But this set of blocks really means that the environment is highly mutated in all these cases of COVID, severe COVID, compared to cases of COVID that are not severe. So let me show you some examples of these metallocytes. Again, remember that this locus here is very interesting. We have not discovered this. It was discovered previously by others, but our method should also pick it up. And here is the result. This is the metallocyte in the region of chromosome 3. This is the region of chromosome 3, about 600 kilobases. And these are all the blocks in here in the dots, in red dots, that are significant by our means. Interestingly, what you see here in blue is the SNPs, the Manhattan plot, but now very much zoom in. And that would be here the tower previously discovered. And what we discovered is that our hits are not exactly in the tower, but a bit downstream, and include all these blocks, the seed blocks and the neighborhood blocks. That actually matches very nicely all these cytokine receptors that you see here. By the way, they only found CR1, but in our metallocyte, we have many more. And they are tissue specific to a certain extent because they are very much significant in macrophages, B cells, neutrophils, and T cells, not that much in epithelial and endothelial. In fact, how many of these cytokine receptors do we find? Sorry. We find them in all the blood cell types, not all of them. For example, we don't find the receptor number 6 in another metallocyte, but these are the ones that we find. So we are identifying things that are very interesting. Let me show you another case. This is what is around, it's a metallocyte around the TIC-AM2, the toll-like receptor adapter molecule 2 in chromosome 5. This is a quite large metallocyte. It's divided in 3 or 4 parts. And the significant seeds are in these 3 regions. This one contains CCD112, this one contains TIC-AM2, and this one contains ATG12. All of them clearly related to the immune response and signaling in COVID. And they are found, for example, TIC-AM in all cell types. So this metallocyte is found in all cell types. Is this general? So when we take all these genes in these metallocytes, we throw them blindly to see what is enrichment, we find regulation of interferon.
Mark, you have less than 1 minute.
I'm finishing. We do find all of them interferon alpha beta signaling in all these cell types, cytokine receptor, etc. So really this set of genes are really enriched. In many of the sites, we have targets that are relevant for the infection, and I'm not going to go into detail, but they are cell type specific and specific for each one of these parts of the pathways. And also we find several targets that has been already annotated as very relevant for infection by COVID, and here they are. And I'm very happy to discuss those targets with any of you. You guys are the experts in the field for that, so drop me an email if you want to discuss any of these targets, and we have many more that we can discuss with you. This is the work of Juan, as I've mentioned before. This is the team, very beautiful people and very nice people to work with. And finally, this is a collaboration, again, mentioning with the Lau Lab, Stadhouder Lab, and the Graph Lab. Thank you.
Thank you very much, Mark. Excellent tool. So I would be here listening you maybe one complete talk. We have a question by Carlos Ciutat. How many SNPs have you determined to construct the latest Manhattan plot, October 2020? Could you construct a sheet to predict the susceptibility to be infected or to react severity in front of the disease?
Well, the number of SNPs is very large. So they have imputed millions of SNPs in that data set. The tower, specifically in chromosome 3, is a bunch of SNPs that are in equilibrium, in linkage equilibrium. They find it in the particular region that is only a few kilobases away of the metallocyte that we identified in 3 dimensions. So these are quite dense data sets. The interesting thing is that when you put them in 3D, you forget about the SNP, individual SNPs. We don't care about any particular SNP. We care about the environment of the SNPs around this 3-dimensional conformation in the cell in particular. So hopefully that answers your question.
Okay, so, well, thank you very much, Mark. As we are very late, I'm so sorry,
yes.
But we have to move to the next talk. So, Juana,
thank you.
So the next talk is presented by Jordi Rodon from the IRTA-CReSA, and the title is Search of SARS coronavirus 2 inhibitors and their combination within approved drugs to tackle COVID-19 pandemic.
So I'm going to present this work called Search of SARS coronavirus 2 inhibitors and their combinations within approved drugs to tackle COVID-19 pandemic. This was done at IRTA-CReSA in collaboration with our partners in IRSI-CAIXA and Barcelona Supercomputing Center, and supported by the financing of Grifols. So as a generalities, just to remind you, and you all know this, that now more than 44 million cases have been confirmed of COVID, including 1.1 million fatalities. Here in Spain, we are in really bad numbers. 1.2 million cases already confirmed and more than 35,000 deaths. As you know, also the main symptoms range from asymptomatics, from fever, cough, shortness of breath, or acute respiratory stress syndrome, to renal failure, septic shock, and it can lead to death. I want to show you this map here, and in a different view. This is the map that I was showing in the first coronavirus symposium, and I was presenting some numbers, and this is the cases of COVID-19 per million. As you can see now, they increased rapidly, and we are entering, especially in Europe, to a second wave. And here, France, Spain, we are in really bad numbers. So what this really means is that time is really matters, and we need to find treatments to cure the patients. So far, only remdesivir has been approved by the American administration, but also there are no commercially vaccines. So in our labs, what we proposed is to compare currently available drugs to counteract SARS coronavirus 2 induced cytopathic effect in vitro, and to prioritize the antiviral activity of these drugs to provide an evidence for coming clinical trials in the hospitals. Okay, so the main antivirals can tackle the different steps of the life cycle of the SARS coronavirus 2, being the entry, either the protease or the replicase of the SARS coronavirus 2, or even the encapsulation or release of the virus. Here, what we did is to differentiate entry and post-entry inhibitors, because entry inhibitors can be used as a potential prophylaxis and also therapy, but post-entry inhibitors are mainly used as a therapy. What is also important, I want to let you know that we are seeing that SARS coronavirus 2 can entry via endosomal route or fusion in the membrane protein, depending on the cell type that we are talking. For example, in the renal cell type, the main entry is via the endosomal pathway, while in the lungs is mainly by fusion in the membrane. So what we did is to get a sample from Canruti, from an infected patient, and then we isolated the virus in the lab. This was done in IRTA-CReSA under BSL-3 conditions, of course. And we further sequenced this virus to know and fully characterize the isolate that we had. And we developed a serial of assays using different cell lines and pseudotype virus or live SARS coronavirus 2 to discriminate between replication and, let's say, entry in using one route, endosomal route, or fusion route. And then we also used pseudoviruses to discriminate if an antiviral was working at the entry stage or a post-entry stage. So what we did mainly is to see different cell lines, VEROI6, CALU3, or different HEK expressing AC2, so the receptor for SARS coronavirus 2, and also another cell line that was expressing TMPRSS2. This is the enzyme that cleaves the spike protein at the membrane. So when we were using this cell line, we were rescuing the activity of this enzyme, and we were changing the tropism of these cells from an endosomal route, only having the AC2 expression, to a membrane fusion using the TMPRSS2. And then we were combining the antiviral with the SARS coronavirus or pseudotype. And after 3 days, we were assessing the percentage of cell entry or cytopathic effect by luciferase. So what we did was mainly to assess for 72 drugs and 28 of their combinations for their capacity to inhibit the SARS coronavirus 2 replication. And among these drugs, we used 16 compounds that were supposed to inhibit viral entry, 22 drugs that were thought to block viral replication, but in the sense of the SARS coronavirus 2 fusion. And then also 11 candidates that were predicted to inhibit proteases and others that were unknown. And this is a work that we are continuously doing in our lab weekly, and we are testing more compounds. So many of them didn't work, unfortunately, but also we found some that they were working at really high concentrations. And for example, lopinavir and tripanavir were two of the protease inhibitors that you can see in this table that they were working, but at high concentrations, more than, well, a bit less than 50 micromolar. And then I want to show you this table with the compounds that they were working in a really low or at micromolar scale. And then we have some also, these three first in green, plitidepsin, MDLs, and NPOs that they were working at nanomolar concentrations. So these are really interesting. And the plitidepsin, the first one, or also called aplidin, is the one working at nanomolar scale and having, well, it is the only one which is approved and being already used for other treatments. The other two are already in preclinical stage. So I'm going to focus now only on these ones. And if we see the entry inhibitors of the SARS coronavirus 2, we saw the hydroxychloroquine. This is the really famous one, which everyone already knows about. And also NPOs and this MDL28 170. These are calpain inhibitors, and all of these had a micromolar activity or even nanomolar activity in some cases. And then fenofibrate, which is also used for other purposes, was showing some antiviral activity. at more or less 20 micromolar. So we had the doubt on which entry routes were these compounds inhibiting, and then we used chloroquine as a main drug for the chloroquine derivatives, and also MDL for calpain inhibitors drugs and fenofibrate, and we assayed with pseudoviruses and cells expressing only AC2 how they work, and we can see that the entry of the pseudoviruses was inhibited using these cell lines and these compounds. But when we transfected the cells with the PRSS2 and we recovered the capacity of these pseudotypes to fusion in membrane, we saw that chloroquine and also MDL and calpain inhibitors were not working against this route. So Camostat was used here as a positive control because it was already reported that this drug is inhibiting the TM-PRSS2 activity. So what we could say is that these drugs are inhibitors of the SARS coronavirus 2 endolytic entry. And then we also looked for post-entry inhibitors, and we could find antiviral activity for the protease inhibitors lopinavir and tripanavir, and also the remdesivir is the one that is approved. We used it and also confirmed their activity against the polymerase of SARS coronavirus 2. But here what we found is also plitidepsin, or also known as aplidin, now is also in all the news, was the compound working in VERO-E6 cells better that is approved, as I told. And this is an antiviral that has been used. And what we were also interested is to assess if this plitidepsin could have any harmful effect or beneficial effect when combining with other drugs. And we found, for example, combining with hydroxychloroquine or either with remdesivir, that this drug was not increasing its effect that comparing to alone, but also it was not decreasing the effect. So since the main therapies or the best therapies sometimes combine several drugs, so this is a rationale that could be used for other clinical trials that are coming. And then what we did is also to confirm these results by ELISA. We collected the supernatants of the cell culture, and we assessed for this activity. And as you can see, also the replication was inhibited with these drugs, with being aplidin also the most efficient, and we confirmed the activity. And to finish, we also saw that interferon alpha and gamma had some activity in VERO-E6 cells. We also collected the supernatants, and we saw inhibition of the replication at some doses. So this is all. I want to thank you for your attention, and this was done in the consortium of IRTA-CReSA, Barcelona Supercomputing Center, and IRSI-CAIXA, mainly financed by Grifols. And I want to thank the team of research of this coronavirus, and BSL-3 technicians and animal caretakers in our labs that are working very actively with us. And if you have any questions, just let me know.
Thank you for your presentation, Jordi. I just have a question. So one problem is like many drugs, they work in vitro, but then they don't work in vivo, no? So we have the chloroquine being one example of how much work was done with this and loss of time. So I understand from your presentation, it's very interesting that trying in different cell lines with different properties, you can discriminate among more, no, a little bit more.
Mm-hmm.
So in this case, for instance, chloroquine went out when you have this AC2 with TMPR as S22 cell line. So my question there, because I didn't see it, how, or maybe you saw it and I didn't realize, how the remdesivir will stay with these two different cell lines, because it's another drug that actually is the only approved, but it's not so clear the benefit.
Yeah, so remdesivir
lines.
So remdesivir works inhibiting the polymerase of the SARS coronavirus 2. So it is not really affecting the entry through one route or another, but acting directly inside the cell, inhibiting the polymerase of the SARS coronavirus 2. So probably this is why it is working and maybe not other drugs. And this is the mainly reason that we didn't try it in the others, because the other cell lines that we used was to discriminate entry, let's say in one route or another, but not for drugs that are working in a post-entry effect.
So in this case, of course, it will be great to discriminate for other steps, like as you say, replication. My question was a little bit in that direction. Do you envision any other way to improve that our results in vitro reflects better what it will be the function than in vivo, maybe with organoids, first test in organoids, or what do you think about it?
Yeah, sure. Yeah, sure. Working with other cell lines or even explants, ex vivo culture, or even organoids could be great. Could be a good way to improve our study and to show how it really happens, the entry and how it works an antiviral in vivo, or how it should work in vivo. But it's true that by using our cell lines, we can also discriminate which routes it works. And for example, if we know that in lung cells, it enters by fusion, while in others by endocytosis, by these in vitro approaches is a good way just to discriminate and first screen the drugs, but we could for sure move to other cell types. Sure.
Okay. Thank you very much again. So we are a little, the time is running, and so the next set of presentation will be like these flash talks, and will be presented by my colleague.
Yeah, okay, let's move to this flash talk. You have 3 minutes. And the first speaker is Jordana Muñoz-Basagoiti from IRTA-CReSA, and she's going to talk about cellular models for the study of SARS-CoV-2 pseudoviral entry. So please, Jordana.
Hello. First of all, thank you for the opportunity to talk here. And I'm going to say that this piece of work is related to the work that Jordi Rodon previously talked about, as we are working together in this big consortium to study antivirals for the SARS-CoV-2. So as you all know, there's no approved treatment for COVID-19, and it is urgently needed. Therefore, there's a call to understand the viral mechanisms involved in cellular infection to make use of already approved FDA compounds tackling specific steps of the viral life cycle. In our lab, we've developed a luciferase-based assay as a tool for the specific study of compounds inhibiting the SARS-CoV-2 entry into susceptible cells. To produce these pseudoviral particles, a co-transfection into HEC293 T cells was done with 2 plasmids, one containing an HIV-1 genome with a defective gene for the envelope protein and with the luciferase reporter gene, and another encoding the SARS-CoV-2 spike protein. So once produced and inoculated onto susceptible cells, these SARS-CoV-2 pseudoviruses produce luciferase upon membrane fusion, so we can study the effect of repurposed drugs affecting specifically the viral entry step. SARS-CoV-2 enters cells via at least two main routes. The first one is the endocytic pathway, led by the binding of spike to ACE2 and its priming by endosomal proteases such as catepsins or furins. And the second one is the plasma membrane fusion pathway, where the spike protein is cleaved by the cellular transmembrane protease TMPRSS2 on the surface of targeted cells, followed by fusion and entry. So when we inoculated SARS-CoV-2 pseudoviruses onto a renal HEC293T cell line overexpressing ACE2, we observed that the TMPRSS2 inhibitor camostat failed to abrogate viral fusion, but chloroquine and inhibitors of catepsins did, as you can see on the plot, indicating that the endocytic pathway was the main route of entry. Nevertheless, pulmonary cells express the protease TMPRSS2, and the fusion membrane entry route is critical for SARS-CoV-2 in them. Therefore, we transfected a plasmid encoding the protease for its expression in 293T expressing ACE2 cells to have a cellular model for the study of the plasma membrane entry route, and observed that in this case, only camostat was effective at stopping the SARS-CoV-2 pseudoviral entry. These results may explain why, in previous findings, chloroquine failed to stop viral entry in pulmonary cells. Following with the previous talk made by Jordi Rodon on compounds affecting the wild-type SARS-CoV-2 replication in vitro, we made use of the SARS-CoV-2 pseudoviral entry assay to corroborate that some of them specifically tackle the entry step. So we could confirm 12 out of 17 compounds tested as promising candidates to specifically decrease SARS-CoV-2 cellular entry, these being cholesterol-depleting agents, chloroquine derivatives, catepsin inhibitors, and the broader cellular protease inhibitor alpha-1 antitrypsin and camostat. So overall, our results highlight that alternative routes govern SARS-CoV-2 viral entry, and these pathways vary depending on the cellular target. Therefore, effective treatments may need to block both endosomal and plasma membrane routes to fully achieve viral suppression. So I would like to thank this amazing group of people, conformed by IRTA-CReSA, the Barcelona Supercomputing Center, and IRSI-CAIXA, and this piece of work was funded mainly by Grifols and John Curono. Thanks.
Thank you very much, Jordana. I have a question. You have heard that now they have found a new cofactor for viral entry, which is the neuropilin 1. Do you think that is going to change your research, the use of this new cofactor?
I haven't had the opportunity to read about this. We've been very busy in the lab, but sure, if there's another route of entry for the virus, we will need to think about other compounds and can stop this, this new factor. And personally, I think that the best treatment we could use against this virus is a combination of antivirals targeting different factors to prevent the entry of the virus by all these different routes.
Okay, but you're proposing most of your antivirals are blocking cellular functionalities. So maybe what about the secondary effects on the patients?
We haven't, well, all the compounds that we've tried in our assay are FDA approved or are in preclinical assays. So in theory, they used at certain concentrations, they won't produce major side effects. And also in our assays, we've performed assays on the cytotoxicity of these compounds on the cells we are using. So we know that the concentrations we are using and in which we are seeing effects on the stopping of the viral entry don't cause major side effects on the cells we are using.
Okay. Yeah, last fast question.
Okay.
Is can the pseudotypes be handled under BSL-2? Stefan Pölhmann?
I would say that yes. Currently at IRSI-CAIXA we are in BSL-3 conditions, but the system that we are using only enters to the cells, so it doesn't have the viral proteins to be able to egress from the cells and reinfect other cells. So I would say that yes, it's completely safe to work with it under BSL2 conditions.
Okay, thank you very
I
much.
have one last question.
Ah.
So the one of the cell line more used in coronavirus research is VeroE6. So this cell line has both types of entry, or which one?
VeroE6 expresses basal amounts of ACE2. So this is why it can be used with the virus. It can produce lots of viruses, but in our assays with the pseudovirus, we found that we need to express the TMPRSS2 protein, this protease, in order for the pseudoviral assay to work. So I would say that we don't see viral entry in our pseudovirus assay when we use this cell line. So this is why we are not using it. Instead, we use these 293 T cells overexpressing ACE2 and the TMPRSS2 in some cases to study the other kind of fruits for the viral entry.
Thank you, Jordana. So the next one is Gerard Duarte, membrane integration and topology of SARS-CoV-2 envelope protein from the Department of Biochemistry and Molecular Biology in the Institut Universitari Biotech Med from Valencia.
So good morning to everyone. My name is Gerard Duarte. I'm a PhD student at the Membrane Protein Group at the University of Valencia. And I'm going to talk about the membrane integration and topology of SARS-CoV-2 envelope protein. Some weeks ago, we published a paper in Open Biology with the topology of the SARS-CoV-2 envelope protein in eukaryotic membranes, showing an N-terminal out, C-terminal in topology. In this case, the experimental design was prepared taking advantage of two native glycosylation sites that the protein has at the cytoplasmatic region. After that, analyzing the sequences from the 7 human coronaviruses, we realized that there is a really strongly conserved positive charge at the end of the hydrophobic region in all of them. And the first thing we thought was that this may act as a topological determinant. In SARS-CoV-2, pay attention to the position 38. This is the argine we are talking about in this case. Apart from that, analyzing the sequences, it's possible to observe that from MERS to SARS-CoV-2, there is an evolutionary tendency to accumulate a positive charge balance towards the cytosol, suggesting that in this case, the insertion responds to the positive insert rule, and probably with multiple determinants, especially in SARS-CoV-2. To study the contribution of these charges and also the role of the arginine 38, the conserved one, in the topological determination, we started some experiments with mutants. The first mutant we tested was this one, in which we replaced 2 negative charges with 2 lysines, 2 positive charges. This mutant was tested in the paper with no effect on the topology, so we decided to try with another mutant, in which we replaced the arginine 38 with a negative charge. In both cases, we still have a positive equilibrium in the inner part, so we decided to finally try a third mutant, in which we combine both mutations. In this case, giving a charge balance of 0. Finally, after testing these mutants in vivo in HEK cells, we saw that all the simple mutants show the same topology as the wild type, but when we combine both mutations, we saw the reverse topology. To confirm these results, we prepared an envelope protein variation with 2 extra glycosylation sites, one in N-terminus and another one in C-terminus, in order to have positive results in both topologies. And again, in this case, we observed the wild-type topology in all the simple mutants, and only when we combine both mutations, the reverse topology shows up, suggesting the strong multifactorial nature of the topology determination of this viral protein, but also the importance of the arginine 38. And that's it. If anyone have a question, I will be glad to answer it. Cool.
Thank you very much, Gerard. Have you tried to find? Sorry. Juana, do you have any question? Okay. So, is there any question? So, is it possible? Have you found? Have you studied whether these mutations can be found in the patients? Or
No, not really. These are totally artificial mutations that we just prepared to study the topology determination.
So this will change the relationship or the binding of this protein with the PDC that Luis Enjuanes has told this
yes,
morning.
For sure.
So
we think that these residues are really important to the topology determination and the orientation in the membrane. So for sure, these are important for the relation with this protein with the other ones in the membrane. And
okay.
Probably it will affect on the packaging also.
Okay, so if there is not any other question, so thank you very much, Gerard. You have to move to the next one because we are 20 minutes late. So let's go to Marco Brustolin. Okay, let's see if you can share and work the microphone.
I'm sorry for the technical issue. So share this. So as Julia introduced to you before, we have different animal models for we have different animal models for the study of SARS-CoV-2, and for some of them, they have already been done study about reinfection. However, they have been done always using the same viral strain between the two challenge. Here we want to show our model is a golden hamster model in which we use two different viral strain between challenge and re-challenge. Here you have the two strain that we use, and you can see that the most llamative difference is the spike protein mutation D614G. So when we infect the animal, the first thing that we have done is follow the weight variation a long time. During the first infection in panel A, you can see how infected animal exposed to CAT01 strain lose progressively weight till day 5, and then they start recovering weight. When we re-challenge this animal at day 21 post-infection with the same strain or with the different strain called Washington 1, we didn't notice any difference or any variation in the weight compared to the negative control. Then we move on and we analyze the viral loads in different target organs by RT-PCR, targeting both genomic and subgenomic RNA level, and also by titration of infectious viral particle in a virus cell. What we see is that we were not able to detect subgenomics RNA in the lungs, and we were not able to isolate infectious viral particles from these organs of reinfected animals. However, when we analyze viral load in nasal turbinate, we can detect SARS coronavirus 2 from both strains from the nasal turbinate at day 2 post-infection. Finally, we analyze the level of immunoglobulin from against 3 different proteins: spike protein, receptor binding domain, and nucleoprotein. And what we see is that animals start to develop immune response at day 7 post-infection. At day 21, before the re-challenge, they have significantly high level of immune response, and this immune response increase at day 2 post-reinfection and day 4 post-reinfection. To conclude, we can say that reinfection is possible with homologous and heterologous SARS-CoV-2 strain. Animal develop protective but not sterilizing humoral response starting at day 7 post-infection. And finally, the lower respiratory tract of reinfected animals showed no evidence of infection. To conclude, I want to thank all the people that are part of this consortium and that make possible this study.
Thank you. Now, I don't have any questions.
Yes, I see one question by Juan José López Moya. How different are the two strains used in the whole genome and in relevant proteins?
The principal difference is the most remarkable one is that mutation in the spike protein that has been related with a more infection capacity of the strain. However, as you see in the presentation, there are over mutation that difference this strain. However, some of them have not been characterized yet. So they have been described but not characterized the effect yet.
So your results quite confirm what we are seeing in patients, that the reinfection causes a milder COVID-19 infection disease?
So what we see in this animal model is that, clinically the animals are less affected during the reinfection. They didn't lose weight. And also seems that there is a protection of the lower respiratory tract. This doesn't mean that the animal didn't get infected, because we detect infection in the upper respiratory tract, and we cannot discard that the animal is still able to shed virus.
Okay, so thank you very much, Marco. Juana,
Thanks.
Let's close the...
We'd like also to thank all the presenters from this session, that it was really interesting. And now actually, principal, we have 20 minutes rest that now we have lost. So Nuria, tell us what to do.
Yes, I think that we just shortened the break and we have only 5 minutes break, if you don't mind.
Great.
Yeah, see you in 5 minutes.
5 minutes. Okay. So 11:30, okay?
Yeah, okay. Thank you.